Alright guys, hey, I'm going to walk you guys through this animation on protein digestion absorption involving all the enzymes and different interactions that occur. It can get kind of complex, so I figured if you have me there to kind of help explain some of the stuff, it might be kind of handy. I may just confuse you even more, but hopefully not. So here we go. Protein digestion and absorption. This is an animation from a website similar to the carbohydrate and the fat digestion animations. I've got several of them, but we're going to take a look at this one first. It's got to load though.
Protein digestion takes place in the stomach and small intestine.
That's important information. Sorry, we had an announcement there. But that's important information because one of the things I expect you to be able to do is tell me when and where along the digestive tract proteins are digested, because these macronutrients become digested in different places. So like I said, protein digestion starts in the stomach and the small intestines.
A stomach enzyme initiates the process by fragmenting proteins into peptides.
There's a function right there. You may need to replay it, but it tells you what pepsin does. There's the first enzyme that comes into play during protein digestion.
Pancreatic and intestinal brush border enzymes complete the digestive process. In the stomach, proteins are initially met.
Okay, time out. The stomach, this doesn't go into a whole lot, but the stomach lining is actually composed of two different types of cells. You have parietal cells and you have your chief cells. I've got another animation, I think, below this that kind of talks a little bit about this together. But again, the parietal cell's responsibility is to create hydrochloric acid, and the chief cell produces the protein pepsinogen, which is, she's getting ready to tell you, is an inactive form, meaning it's not ready to do its job yet. It's not until these things get combined and pepsinogen turns into pepsin. Remember, when molecules change shape, they're recognized differently. So due to that change in shape and that they become activated and have that active site necessary for breaking down these proteins.
Of pepsin. The pH lowering effects of hydrochloric acid convert pepsinogen to pepsin.
We talk a lot about homeostasis and maintaining a balance. These enzymes actually happen to prefer and work better at a more acidic pH. So that's one of the reasons hydrochloric acid is released in response to these proteins coming in. So your body can digest them properly and provide the conditions in which these things are designed to work.
Pepsin molecules, in turn, act to catalyze the production of even more pepsin.
Over here, interpreting diagrams, understand the little white spheres, the hydrogen ion, and you get the pepsinogens, that's the enzyme, or the protein, and the chloride ion, because when the hydrochloric acid will dissociate or break apart into these two different ions.
The catalytic action of pepsin breaks proteins into fragments called peptides.
Again, they're called peptides, named after the type of covalent bond that holds them together. The amino acids are held together by covalent bonds called peptide bonds. So they call them peptides. Sometimes they call them polypeptides, dipeptides. I guess you want to get real technical, tripeptides and illegal peptides. It all just kind of like we talked about before. Those prefixes just determine the number of amino acids in the chain. All right. We'll click on a peptide here.
Protein digestion continues in the duodenum of the small by a digestive mixture called pancreatic juice.
All right. So once the food leaves the stomach, it goes in the first section of the leaves the stomach, goes in the first section of the small intestine called the duodenum or duodenum, depending on who you talk to. And this is that liquidy stuff that's basically digested food is called chyme. I believe it'll come up here on the right side here in a minute, but it's spelled C-H-Y-M-E, chyme. And right there is about the point when the gallbladder and the liver and the pancreas all start getting involved, because they got these little ducts here that dump their necessary secretions and juices into that area so they can start mixing and then travel along the digestive tract and do their job.
Contains the protein digesting enzymes trypsin, chymotrypsin, elastase, and carboxypeptidase.
Okay, we're getting into some technicality here. So there are basically three different types of four, I believe. I stopped too soon. Different types of enzymes are going to be involved in breaking down these things.
Each enzyme effectively breaks a particular peptide bond to produce smaller peptides or amino acids.
So summing it all up, putting it all together, these things are what break apart the proteins for digestion, and they have specific types of proteins depending on the molecular structure that they're breaking down. I told you they're very, very specific, shape dependent. So you got one certain enzyme that's going to attach to the carboxylic acid side and break a bond. The other side, other proteins attach other places and break those bonds.
Trypsin, chymotrypsin, and elastase work to catalyze the breakdown of larger peptides into smaller ones. Carboxypeptidase breaks the peptide bond that attaches the terminal amino acid to the carboxyl end of the peptide.
Okay, terminal amino acid, that just means the end, all right? So this carboxypeptidase, all right, again, carboxy because it attaches to the carboxyl group. I told you they're named after what they do a lot of times. Peptidase, they break a peptide bond, and the suffix ase means it's an enzyme. So this thing chops off the end, the terminal end amino acid, and a lot of times creates dipeptides, tripeptides, or just free amino acids depending on the size of after the trypsin and the chymotrypsin and elastase have broken them up in these smaller fragments. All right, let's click on a small peptide. There we go.
Protein digestion is completed in the brush border cells of the villi. Two brush border enzymes are involved, aminopeptidase and dipeptidase.
Okay, time out. Aminopeptidase and dipeptidase. You might be able to infer based off their name what they do. One of them is going to involve the free amino acids, all right, and the other is going to involve these dipeptidases, all right. Again, the peptide, this normal peptide will come into play here in a moment as well. It's called the brush border because you can see these finger-like extensions, the cilia, if you want to call them, look like a brush.
Catalyzes the breakdown of peptide bonds that attach the terminal amino acid to the amino end of the peptide. Dipeptidase breaks the peptide bond that holds two amino acids together. The digestion of proteins produces amino acids, dipeptides, and tripeptides.
So summing this big picture all up, these things got to be broken down into very, very, very small fragments, either in 3, 2, or 1 amino acid chains. Again, this aminopeptidase has a similar job as a carboxyl peptidase as far as breaking off the terminal end of an amino acid, a single amino acid. Click on our product.
The end products of protein digestion, amino acids, dipeptides, and tripeptides, are absorbed by the villi of the small intestine. Proteins are absorbed into the bloodstream via 3 mechanisms.
All right, we're getting ready to get into the 3 types of cell transport. I'm going to lump them all together in the same category based off how they get across, but just pay attention to this.
Transport, and both sodium-dependent and hydrogen-dependent secondary active transport. Most proteins are absorbed as amino acids via active transport processes in the duodenum and jejunum.
Again, active transport is when things have to use ATP because they're active. To be active requires energy, so that's why they name it. It's ATP dependent movement. So in order to get across membrane, it's going to require energy from the cell to get it across. And a lot of times it's because that energy or that molecule is being moved against the concentration gradient, or in other words, the cell may already be full of them and is able to continue moving it across there. To keep filling it up. Sometimes they call it going uphill, going from low to high concentrations rather than going from high to low like in passive transport. So, which is another reason why it needs energy. It needs energy to move it across the membrane and keep packing it in. And another reason why they call it active transport.
They also enter epithelial cells via sodium-dependent secondary active transport. Dipeptides and tripeptides are symported with hydrogen ions.
Okay, sodium dependent, not going to cross the membrane without the addition of sodium. All right, If sodium's not there, they can't move across. So it's sodium-dependent active transport. And then you've got this other method of sim porting. Sim means the same or at the same time. So in order to get across, they have to have hydrogen going along with it in the same direction. There are things called anti-porters, which this doesn't show. The anti-porters move things in opposite directions. So something may come in on this side, something comes on this side, and they switch places. This one will go through, and this one will come through in the other direction. That's called an anti-porter. This is a symporter where they're both going in the same direction at the same time.
Once inside the epithelial cells, the peptide compounds are hydrolyzed to single amino acids. In the final stage of absorption, amino acids move from the epithelial cells across the basolateral membrane through the interstitial fluid and into the capillaries via diffusion.
Okay, here, so in order to get from the intestine into these border cells, they've got to go through active transport. They use energy to get across, or sodium or hydrogen. Once in here, they're broken down further and make sure everything's an amino acid. There are no more dipeptides or tripeptides or anything. Everything's amino acid. And from there, they can facilitatedly diffuse. I think I just made up a word. They can diffuse using transport proteins, in other words, to get across. Remember, amino acids have a charge on them, so in order to get across that nonpolar barrier provided by those phospholipids, they've got to use a protein bridge or channel to get through. But it doesn't require energy this time because there's a ton of them in here, and they're all bouncing around, ping-ponging, and eventually they just kind of diffuse out, and they cross this interstitial fluid, which is the fluid between cells and tissues, and pass over, and then they will diffuse because there's a high concentration of them into these blood vessels and capillaries for transport where they need to go. All right, that sums up protein digestion. All right.
Hallway. If you haven't made it to the auditorium, please come.
I apologize for the announcements, but pause it, rewind it, do what you need to do to help you understand this a little bit better. But again, pay attention. When and where do those proteins become digested?
Anyone who has not been dismissed, please come to the auditorium at this time.
And how? What enzymes become active, and what do they do? Guys, best of luck. Of course, if you have questions, let me know.